Have you ever wondered how your body is able to carry out such complicated processes like growth, maintaining a temperature, or metabolism? Or maybe you've been curious about that butterfly-shaped gland at the bottom of your neck. If so, you're in luck. Today I'm going to tell you the origin story of the thyroid and all about the enzyme who made this possible. Before we travel back in time, let's take a look at the modern-day thyroid. The thyroid gland is a major component of the endocrine system and is located in the bottom half of the front side of your neck. It plays a critical role in regulating metabolism, growth, and development through the secretion of thyroid hormones. Today, the thyroid is regulated by the hypothalamus-pituitary-thyroid axis, or the HPT axis. In the brain, the hypothalamus releases thyrotropin-releasing hormone, which stimulates the pituitary gland to release thyroid-stimulating hormone, and then finally stimulates the thyroid to release T3 and T4 into the bloodstream. These superduo teams, composed of monomers monoiodotyrosine and diiodotyrosine, are the primary hormones released from the thyroid and are responsible for regulating various physiological processes throughout the entire body. From metabolism to growth and development to the nervous and reproductive systems, hormones T3 and T4 are crucial to the function of all. Basically, they're kind of a big deal. We'll get back to these guys later in the story. Cool process, right? But how did it all start? Let's introduce the enzyme who made it all happen. Once upon a time, there lived an enzyme named thyroid peroxidase. He liked going by TPO for short, so we'll call him that for the rest of the story. TPO was your average enzyme. He grew up in the thyroid, liked working out to make sure he didn't denature, and loved to watch his favorite TV series, Breaking Bond. However, TPO had big dreams and always had a desire to help out other enzymes and amino acids all over the body. So, TPO attended Blandford University in the thyroid. It ended up taking him 6 years to get his degree because he could never remember the Krebs cycle when it came to his final exams. But he did get there. One day, shortly after TPO's graduation, a strange man approached him on the street. His name was hydrogen peroxide, but TPO called him Dox Perox. Dox Perox saw the young spirit and intelligence in TPO's eyes and asked him if he'd want to help start up the thyroid's first ever optometry clinic. Dox Perox explained how a lot of the amino acids living in the thyroid are usually visually impaired, especially the tyrosines. Because they are visually impaired, non-functional tyrosines can cause a lot of problems and are never able to function properly or live to their full potential. He knew he could help them, he just needed an enzyme that knew how to oxidize eyes. TPO was hesitant at first, but he did learn how to oxidize eyes at Blandford. So, he agreed to join Dox Perox in business and started up the first ever thyroid optometry clinic. From that moment on, Dox Perox and TPO helped little tyrosines all over the city become able to see and therefore fully functional. Let's look at the biochemical mechanisms that TPO and Dox Perox use that allows tyrosines to see. As mentioned before, thyroid peroxidase is a major enzyme in the biosynthesis of thyroid hormones. TPO has catalytic activity for two substrates and is responsible for both the iodination and the coupling of tyrosine residues located in thyroglobulin, a glycoprotein which generates the hormones triiodothyronine, T3, and thyroxine, T4. Let's take a closer look at the structure of the TPO enzyme. Thyroid peroxidase is a 933 residue transmembrane homodimer enzyme anchored to the apical membrane of thyroid follicular cells in the thyroid gland. It is made up of several domains, as shown here, but we will be focusing on the heme-containing domain located in residue 238 of the 933 residue enzyme. TPO contains a heme group in its active site, which is a ring-like organic compound known as porphyrin, to which an iron atom is attached. It is this iron atom that reversibly binds to oxygen. As the TPO enzyme is a heme peroxidase, it can't oxidize the substrate without first being oxidized itself. That's where hydrogen peroxide comes in. One oxygen from the hydrogen peroxide binds to the iron atom in the porphyrin ring of the active site, resulting in the oxidized form of TPO and a water byproduct. The exact mechanism by which hydrogen peroxide oxidizes TPO is heavily debated and is still being researched today. However, it is proposed that the resting state of iron 3+ is oxidized to the highly reactive form of iron 5+ double bonded to an oxygen, a species that is formed in many systems containing porphyrin rings. TPO is now ready for the iodination and the coupling of tyrosine residues in thyroglobulin. This next mechanism by which TPO turns iodide, the form of iodine found naturally in the body, to the iodonium ion, the form of iodine that binds to tyrosine, is also not fully understood. The specific domain within TPO that initiates this process also remains unclear, which is super inconvenient for the point of this video. But once TPO does convert iodide to the iodonium ion, they are ready to be added to the tyrosine residues in the thyroglobulin protein. When TPO only adds a single iodonium ion, monoiodotyrosine is created. When he adds two, diiodotyrosine is created. Let's zoom in on an individual tyrosine to see how this process happens. First, the pi bond between carbons 2 and 3 in the aromatic ring on the side chain of tyrosine will be our source of electrons. Therefore, it is the nucleophile in this first reaction. The electrons grab the iodonium ion, creating a carbocation intermediate that is resonance stabilized. Deprotonation occurs to restore the pi bond between carbons 2 and 3 to give us our product, monoiodotyrosine, or MIT. To form diiodotyrosine, the product from the first reaction, monoiodotyrosine, goes through the same process as before. The pi bond grabs the iodonium ion and forms a carbocation intermediate, and then undergoes deprotonation to restore the pi bond between carbons 4 and 5 this time. The product, DIT, is formed. In the last step of thyroid hormone synthesis, the reaction between MIT and DIT, or two molecules of DIT, is catalyzed by TPO. Oxygen will act as a nucleophile, and the chain containing the amino and carboxyl groups will leave as the leaving group. This group is normally not a great leaving group, however, can do so in the presence of TPO. Teams triiodothyronine and thyroxine can then travel into the bloodstream and all over the body as they are now fully functional. As mentioned before, T3 and T4 are very important for many systems of the body. And so, that pretty much concludes our story of how TPO and doxperox helped so many tyrosines C and continue to do so today. An important note, however, is that this is not always the case. Occasionally, TPO experiences some issues. Sometimes, TPO gets attacked by antibody bandits. TPO can act as an autoantigen. This means attacks are mediated by the body's own immune system accidentally recognizing the antigens on TPO as harmful. After all, the West Side maybe wasn't the best location choice for their clinic. The antibody bandits can attack TPO and cause him to be injured. Therefore, he is not able to give as many tyrosines the ability to see. This happens in Hashimoto's disease, the most common cause of hypothyroidism. An underactive thyroid slows down many bodily functions and includes symptoms such as weight gain, hair loss, and fatigue. Other times, TPO sees the bandits ahead of the attack, outside the window of the clinic. He panics and starts helping a bunch of T3 and T4 teams, but way too fast, causing chaos. This happens in Graves' disease, the most common cause of hyperthyroidism, or overactive thyroid. When the thyroid produces more hormones than needed, this causes many bodily functions to speed up, resulting in symptoms such as weight loss, tremors, anxiety, and increased heart rate. However, these are only exceptions, and overall, TPO and doxperox do a pretty good job at helping the T3 and T4 teams' vision and allowing them to travel outside the thyroid to do amazing things all over the body.